Hello, I'm Laura Cassidy from the American Chemical Society. Welcome to this news briefing from the American Chemical Society Spring 2019 National Meeting in Orlando. We're joined today by Dr. Jean-François Lutz of Institut Charles Cedron. He's studying on-demand polymers that could lead to novel computing and fraud detection technologies. Dr. Lutz?
Thank you very much. So the paper I would like to speak about this morning is some discoveries regarding what we call information-containing polymers. So I'm a polymer chemist. I work on synthesis of macromolecules. And you may know that there are two types of polymers. Ones are conventional plastics, what is actually having a bad press, usually polluting, and that you find as microplastic in the ocean, for example. On the other extremes, you have biopolymers, like DNA or proteins, which are much more precise polymer with very precise structure and very controlled sequence of information, and this is what is determining life. Actually, what we are doing is maybe to try to find a compromise in between these two extremes, and so in between, let's say, ill-defined plastics and extremely well-defined biopolymers. What we are intending to do, you have it on this slide that you can see on the screen, is to find new methods of chemistry to synthesize polymers which are man-made, which are synthetic, but which are as defined in terms of structures and properties as biopolymers. So the molecule you see on the screen is an information-containing polymer. And as you can see, you have some monomer units. These are the light blue and dark blue balls. And basically, we use a chemical procedure to put each of these monomers at the right place. So like in a DNA or like in a protein, we can write a language. So it doesn't have to be a natural language. It can be something else, like, for example, digital information. On the slide you see here, you have actually 3 times 8 monomers. And this is what is Ascii encoding. So in Ascii, 8 monomers define a letter. So actually the first 8 monomers define the A of ACS, the second 8 the capital letter C, and then S. So on the macromolecule you see here, which is just a view, just a cartoon, we wrote, and we can do it practically, the word SES at the molecular level on a molecule. And so we can write information on polymers, but of course we can also handle this information. So we developed some sequencing method to read and to extract the information from the polymer. Also some methods to edit the information, like, for example, erasing information, things which are quite easy to do with synthetic polymers, but would be more difficult to do with DNA, for example. So this is what we research that we are developing in my lab, and the potential applications are several. One is, of course, data storage, and if you imagine to store information at the molecular level on a molecule, you are at a size which is much smaller than in a typical storage medium. So you may have practical application, although this is a far away dream, I would say. We need still to have many, many years if we could eventually develop some informatic device based on these technologies. A more realistic application, this is the title of the paper we discussed today. This is anti-counterfeiting. You have here a very simple plastic which contains a message. So if you are a company, you can potentially write the name on the company, a batch number, production date, and then you have a very easy molecule that you can process and blend in other materials. That can be in another plastic, that can be in a ceramics, that can be maybe on a metal surface. So this is a little address, a barcode that you may put on many materials for identification and traceability. So we develop many examples of materials that we can tag using this kind of technology, and we believe that there is some potential future, fundamentally speaking, but also in applied chemistry with this kind of molecules.
Thank you, Dr. Lutz. Are there any questions? Please state your name and affiliation before asking your question.
Hi, so it's Kath O'Driscoll from Chemistry and Industry magazine. And the press release talks about DNA as being this very information-rich molecule. I wonder how far some of your molecules actually approach DNA in terms of the information that they contain?
Yeah.
Can
So-
you sort of
so-
give an idea of how much? And
It's an
for
excellent
what
question. We are a bit late as compared to DNA. DNA, there was 4 billion of years to optimize the chemical structure. We are just working on this topic for 4 or 5 years, so this is not the same competition in a sense. And so DNA is, of course, highly optimized by nature to contain information and a high content of information. Our polymers are more modest so far. We can put about, let's say, 100 bits of information on a polymer chain, which is about 10 bytes. This is very humble. But we can imagine, and this is also what people with DNA are doing, basically we don't need to have very, very long polymers containing information. The idea is maybe to have a polymer with 100 units, and then to create libraries of information that we will orientate maybe in 2D or in 3D to have maybe some microarrays which could be used like a chip to contain information. Our polymers got other advantages as compared to DNA. So DNA is highly optimized, as is what I said, by evolution to be a biological polymers, but the application we discussed today are not biological. Data storage or anti-counterfeiting, this has nothing to do with biology. So DNA is very practical to contain a lot of information, but it's also not a very, very robust molecule. If you just have fun to put DNA in an extruder, for example, you will see that it has problem to survive thermal treatment or mechanical treatment. So this is a polymer which is degrading quite easily. Okay, so if you do the same job on synthetic polymers, they may be much more robust thermally speaking, chemically speaking. So this is something we have been demonstrating in several papers for this, let's say, processing of the tag that you want to put. These polymers are much more robust than DNA would be.
And I know with DNA, it's the bases that then code for the information.
Yeah.
And with these, you're saying it's the monomers that do the coding. Can you do it with all polymers as well? Is it?
Exactly. DNA is a polymer. Our polymers are polymer. And the bases are monomers. Okay. The 4 monomers of DNA are ATGC, basically. So you have 4 monomers to create a code. In our case, what is a chance for us is that we are not stuck to these natural codes. We do whatever we want. Okay, so we can take as much monomer as we want. We just require 2. You can take 2 monomers and say this is my 0, the other one is my 1, and then you create your sequence. But of course, we can also work. This is synthetic chemistry, so we have no limit. If we want to have alphabet with 4 digits or 8 digits or 16 digits or 32, we can imagine the chemistry. Okay, but principally speaking, this is very close to the concept of DNA.
Okay, thank you.
Charles Berquist from Science Friday. Could you discuss, say I'm a company, I've created your barcode to say this is batch 312 or whatever. What's the readout process for the guy in the warehouse wants to know what batch it is?
So the main, we have several readouts, and this is a lot of fundamental chemistry. We cannot apply all the sequencing technique that exists for proteins and DNA because there is sometimes some biology involved. For synthetic polymers, the main tool is mass spectrometry. And so typically we use a method which is electrospray. Which permits to read the code quite accurately. We have very few errors in reading. We also developed some softwares which permit to have a very fast analysis. So typically the measurement time will be about some minutes. And then we have a software which permits to decode automatically the polymers, and this proceeds in about a millisecond. So it's something which is quite practical. And there are also some methods of mass spectrometry that you can directly use on surfaces, like DESI, for example, desorption ionization. So this is also an advantage to directly go on the surface of a material and try to find your code. Okay, so this is what we use as the main tool. Mass spec is still a relatively large instrument, but there are now many generations of new mass spectrometer which are table instruments. These are not tested yet, but principally we may also imagine to have field analysis with these kind of things, and that somebody could be with a car basically could still track coded materials using this kind of sequencing methods.
Belo Buslig, American Chemical Society. Do I understand it correctly that you synthesize the biopolymer essentially and splice it into just ordinary polymer of some sorts. And the second question is, are you just confining it to the ATCG pairs, or are you using the newly developed isomers like Stephen Benner has been working on?
So we use fully synthetic structures. They are DNA-like, but they are not DNA at all. So it's not that we use DNA and non-canonical monomers like Stephen Benner's and others. We use materials which contain information in the same way, but which are different polymers. So it can be polyurethanes, for example. This can be polyamides. This can be non-natural polyphosphodiesters. So our polymers are fully man-designed and man-made, basically. Yeah? And so this is, and to answer the other part of your question, this coated polymer, which is of course quite expensive to do because we have to proceed monomer by monomer, we blended it, this is what you said, in some other plastics, but at a very, very low content. So typically we would work at traces, ppm, something like that. So you don't need to have a huge amount of these coated polymers to put it in a material. Think about maybe your cell phone, maybe in the plastic of your cell phone, you want to have a protecting tag. We would put maybe a few ppm of these coated polymers inside this plastic. It won't change the properties. It won't change the appearance. It won't change the mechanical properties. But you would have inside a little label that you can track, identify with mass spectrometry, and which permit to detect and identify your material.
Now, when you've got that biopolymer kind of spliced in there, how do you normally polymers are just a single strand and so forth. Of course, DNA is a double stranded. How do you splice that in there so it stays as pairs?
Okay, so we don't use double helixes. We have single-stranded. This is exactly what you have on the slide. So this is, for DNA, you are fully right. You have double-strand polymers. In our case, our polymers are not self-replicating like DNA. We cannot amplify them like DNA. We use them as such. We write a code in it, we blend in the material, we track the code, and we extract the code, basically. So there is no amplification, no artificial biology of any kind. So basically, we synthesize the single strand, and then we blend in another material. And for this is just polymer physics. As we use very small amount of these coated polymers, it's mixed very, very easily in a wide spectrum of materials.
Thank you.
Hi, so it's Kath O'Driscoll from Chemistry and Industry. And I was just going to ask about the process, because I understand it's an iterative process based on the idea of these peptide synthesizers. How is it adapted from the traditional peptide synthesis method to actually produce?
So this is a good point, because I'm a polymer chemist. And the idea here is not to make total synthesis. I don't want to synthesize these polymers in 2 months. I want to have really a process which looks like a polymerization, something easy, cheap, eventually scalable. So this is the point when you do peptide synthesis. This is kind of time-consuming, and typically to install 1 amino acid, it would last 2 hours or something like that, in the best case. In our case, it's a bit different, because this is what I said earlier. We can select any chemistry we want. We are synthetic chemists. We are not looking for a particular structure from nature. We just want to create a new property. Okay? So we can select chemical reactions, which are helpful. So you can really imagine, so for example, we work with some reaction which proceeds not in 2 hours, but in a few minutes. And also we work a lot on orthogonal pathways, which permit to avoid to have some protection, deprotection, what you know from peptide chemistry, which make the process a bit longer. So the whole idea is to find easy step of synthesis that this kind of polymers can be much easier to synthesize. So we already achieved quite a lot in that regard, but there is many, many things more to develop in that.
And how quickly can you produce these, in what amounts?
I mean, this one, if I use, for example, this minute chemistry that I mentioned, this is something that we can produce in something like half an hour or something like that, yeah. So typically we can make very long information polymers with 100 coded residue in about 6 to 10 hours, which is very normal time to make a polymer. If you make a standard homopolymer or block copolymer, this is also in the hour range. So this is the whole idea, that the chemistry is fast.
And just another question in terms of, because these polymers, the coded polymers are blended in, I just wondered, I know it's in very small amounts, but would it affect recyclability? And would you still get the coding in the recycled product?
Yeah, so this is actually a very important application. There was already a call of the European Union for funding for this, to use this kind of polymers as trackers for recycling materials. So there are some projects starting about that. Typically, you would have big batch of plastic, and you use small amount of these trackers for recycling the materials, for circular economy and this kind of things. You may have something which permit to identify your batches. So this is also an application that I didn't mention, but where we believe that this kind of little molecular address may have a very big potential. Yeah.
Okay, thank you.
Katie Cottingham from ACS. So would these polymers be best for encoding kind of simple messages and little tags, or could they someday be used to store large documents and things instead of paper or a computer hard disk?
Yeah. I mean, so for DNA, so there are many, not me, but other person working on putting information on DNA. The level of storage is already above megabytes, maybe approaching slowly terabytes. So it's already, it's not a data center yet, but we can make some stuff which are already quite a big amount of data. In our case, so you have here something which is 3 bytes. We reported last year some materials containing about several hundred bytes, no, 160 bytes to be precise, meaning that we are flirting with the kilobyte. So we may imagine, of course, to have larger content of information. It's not impossible, but it's also a matter of easiness, of price, and so this is something that we are developing slowly, but there is nothing against the fact to have high amount of storage. Yeah.
And when do you think this technology would be implemented commercially?
So actually for the anti-counterfeiting, and I cannot reveal much, but this is extremely close to market. So many things are patented. Companies are involved. I cannot mention them. So I would say for this kind of application, like tracking recycled materials and identification, this is application, we have maybe a time until commercialization, maybe 1, 2, 3 years. Okay, what really matters at this stage is only price, but not really the technology. Data storage is a faraway dream. And this, maybe it will take another 10 years or 20 years, or maybe the application won't be the one we think about. Maybe I think it will never be an artificial computer based on polymers, but maybe you may imagine archives, if you want to store, let's say, some things that you will not use every day. A big part of what we store, we don't use it every day. So old movies, old books, genetic database. So maybe to store this huge amount of sleeping information in a very small amount of matter, maybe this kind of materials could be useful. But also for this kind of development, you may count 5, 10 years, 15 years. This is more fundamental chemistry for the moment. Yeah.
Are there any other questions over here?
Charles from Science Friday again. You mentioned recycling earlier. Do these sequences survive the recycling process? I mean, is there a danger of contamination that I'd have a product with 8 people's tags in it?
Yeah. So this is something which is not tested yet. As I said, some projects are starting, and it's exactly what should be verified now, that you can survive some different cycle, that you do not lose the information. So this requires a bit of development. We know that when we put, for example, some of these polymers in extruders, they survive the process. How many times? I cannot answer the question yet. We have to test all of this. Yeah.
Hi, Bela Buslig, ACS. Going back to contamination problems with natural DNA pieces and so forth, would it be perhaps an approach to use those isomeric pieces, so there is no way that some natural product ever would
Yeah.
be contaminated.
This is an excellent point, and it's actually one of the big advantage. If you use DNA as a tag, you have a huge problem of contamination because an experimenter may touch the sample, or if you want to use it in biological condition, you may have contamination from other genomes. In our case, our polymer, as we discussed earlier, they are fully different from DNA. They have their own chemistry, so there is no contamination from biology. In that case, we can distinguish them quite easily. They are very different from DNA, so this is, of course, an advantage by itself. Yeah.
Thank you.
It's Kath O'Driscoll from Chemistry and Industry, and it was about the medical implants, because I was surprised that 8% of medical implants, it was saying, are counterfeit. And I understand you're using this technology for ocular implants. Can you just say a bit more about that?
So we even went further, and we went in vivo. There was a paper last year. Actually, there was in France a huge problem with breast implants, you may recall. Many women suffered. There was some case of cancer, I believe, because some companies, they were using fake silicones. And the question is, when you have an implant in the body for many years, and you did not keep packaging, how do you know that this implant is a fake or it's a real one? Okay, so this kind of polymers could be very helpful, and so we have been demonstrating it not on silicone implants, but some model implants which were made with polyvinyl alcohol, and this was a collaboration with the lab experience in implants in Paris, in France, and we implemented these model implants in rats, containing some coated polyurethanes, and this was stored in vivo for 3 months. And after 3 months, we extracted the implants, analyzed the implants by mass spec, and we could still find the code. Okay, so this is something you potentially would have very big difficulties to do with DNA, because putting foreign DNA inside of organism would have maybe some ethical issues. In our case, of course, we have to prove biocompatibility, but with the rats we were testing, there was no sign of inflammation or some problems. Obviously, we were seeing that the labels we had in the implants were not leaching out, were not contaminating the animals. These were very simple animal studies. This was just a very first level of test, but it looked like also in vivo we can apply this material. So this may have some high impact as well.
Thank you.
Thank you. The archived version of this session will soon be posted at bit.ly/ACSlive_Orlando 2019. Please join us for our next press conference at 1 p.m. today on targeting a key protein to treat depression, obesity, and chronic pain. Thank you.